Turns out we have to do when somebody's sick or injured is that we have to start the healing process. To start the healing process requires energy, requires high energy molecules in the cell. To rebuild any structure in the cell, we need energy. To perform any function in the cell, we need energy. To eliminate any dangerous molecules or damaged molecules, we need energy. So energy is required for everything that we do. It's required for gene expression, to make new molecules, to get rid of old molecules, to repair damage. All that requires energy, and 90% of that comes from your mitochondria. Every function of a cell requires energy to perform all these different functions, whether it's moving molecules around the cell, or it's providing energy to make new molecules, or whether it's providing energy to maintain the barrier function and to separate various compartments within tissues and within cells. All of this turns out to be very important. And when these membranes break down and the barriers break down, then things start to fall apart in cells, and this can produce what we call disease. So it's not only important in terms of disease, but it's also important in natural aging, because as we age, these barriers tend to become dysfunctional and tend to break down. And so people are more susceptible to infections, for example. Their immune systems don't work as well. All the sorts of things that go on that are necessary to maintain normal housekeeping functions in cells tend to start to wind down, and then things start to go wrong. Something that I've worked on recently, long COVID, which is a condition where you have the symptoms of COVID that last long after there's any detection of the virus in your system. So one of the things that happens with these lingering, persistent portions of disease is our barrier functions are not being maintained properly. So we're not making the proper energy molecule. We're not maintaining our function in terms of our nervous system. We're not maintaining our gut function and so on. And so for all these situations, membrane lipid replacement is important. Why is it important? We know that there are various membranes in the cell that perform all sorts of different functions. We're always repairing them, night and day, 24 hours a day. We're repairing these lipids. We're bringing in lipids from outside and repairing them, because a lot of this can't be done enzymatically because it's just too difficult to replace these double bonds in lipid molecules. So we have to bring in, it's much easier to bring in new lipids that contain these. So we need a pristine source of these lipids. The mitochondria have to be repaired first. What happens in infections? The mitochondria have to be repaired. And what happens in all these other injuries and everything else that goes on? The mitochondria have to be repaired. So that's one of the primary functions of membrane lipid replacement, is to repair the mitochondria so we can return that intermitochondrial membrane to function. And when it's functioning properly, there's a transmembrane potential across the intermitochondrial membrane of about minus 150 millivolts. So mitochondria are little electrical capacities in themselves. And if that minus 150 millivolt falls, then we can't make the ATP. We can't make the high energy molecule. It's also important in nerve impulses. If the resting potential of the nerve across the plasma membrane, if that potential starts to fall, we can't have nerve transmission. So it damages the nerves. And so all kinds of things depend upon the electrical properties of cells, and membrane lipid replacement helps maintain those electrical properties, which are absolutely essential for a number of different functions. Turns out it's being used for things like heart attack in Eastern Europe, where they give IV membrane lipid replacement immediately after a heart attack. And turns out in their studies, it's very important for recovery from a heart attack.
Hello, everyone. Welcome back to Health Alliance Australia's podcast. My name is Jeannie Rose Angelrother, and I'll be your host today. Today, we have a very special guest who not only is one of the most cited medical researchers, but has a very captivating backstory. It's going to be a very informative interview that will give you a framework for understanding health, with particular emphasis on cell membrane integrity. It also ties into our other important interviews on biowarfare. Professor Emeritus Garth L. Nicolson is the founder, president, chief scientific officer, and emeritus research professor and molecular pathology at the Institute of Molecular Medicine in Huntington Beach, California. He is also the conjoint emeritus Professor of the University of Newcastle. He was previously the David Bruton Junior Chair in Cancer Research and Professor and Chairman at the University of Texas MD Anderson Cancer Center in Houston. And he was the Professor of Internal Medicine and Professor of Pathology and Laboratory Medicine at the University of Texas Medical Center, Houston. Professor Nicolson has published over 700 medical and scientific papers, including editing 20 books, and he has served on the editorial boards of 30 medical and scientific journals and was the senior editor of 4 of these. He is the co-author of Fluid Mosaic Model of Cell Membranes, 1972 Science, now the textbook model for biological membranes. Professor Nicolson has won many awards such as the Burroughs Wellcome Medal of the Royal Society of Medicine, UK, Stephen Paget Award of Metastasis, Research Society, US National Cancer Institute Outstanding Investigator Award, the Innovative Medicine Award of Canada, and the EU Academy of Science. He is also the Colonel Honorary of the US Army and US Navy SEAL Honorary for his work on the armed forces and veteran illnesses. On the scholarGPS international rankings, which are based on productivity, impact, and quality of their scholarly work determined by publications and citations, and H-index, Professor Nicolson ranks number 1 for cell membranes, number 2 for cancer metastasis, number 13 for tumor progression, and number 53 for cell and molecular biology. He is a world authority with a very impressive list of achievements and knowledge. Professor Nicolson also wrote the book, The Project Daylily, a story that chronicles the events surrounding what the public knows as the Gulf War syndrome. To this day, the public perception of that tragedy is very limited. But now there are over 150,000 veterans of that conflict that suffer from chronic illnesses and tens of thousands who have died without acknowledgement or the proper assistance to keep the secret of this origin of their illnesses. Project Daylily is a book that tells the story of the discovery that men and women of the US Armed Forces were actually exposed to chemical and biological mixtures from missiles and sprayers during the Gulf War that were supplied in part by the sinister network using a group of rogue bureaucrats, intelligence operatives, and scientists. They were also exposed to contaminants in multiple vaccines given during deployment. Project Daylily presents the story of how one of these biological agents was found by 2 American scientists and the veterans of the Gulf War and its civilians as part of a massive testing program, and how various academic and government employees did everything in their power to prevent this information from being released to the American public. This is a fascinating story where Professor Nicolson and his wife fled for their lives from Texas. His colleagues were killed and his boss was shot in the head in his office because he was going to expose the biological weapons experiments on Texas prisoners or at Texas prisons. You can see how our interview today with Professor Nicolson ties into our other podcast on bioweapons, in particular, our interviews with the late Professor Francis Boyle, who drafted the Biological Weapons Anti-Terrorism Act of 1989, Dr. Richard Fleming, author of Is COVID-19 a Bioweapon, Dr. Kevin McCann on global emergentic health disaster caused by infectious biowarfare pathogenic program, Elizabeth Glass, and Major Tom Havlin. All of these interviews are available on our channel or via our website, healthallianceaustralia.org. Professor Nicolson has helped many Gulf War veterans recover, and today we are going to focus on membrane lipid replacement, a natural supplement that helps those exposed to various infections and toxic chemicals to recover. Thank you, Professor Nicolson, for coming today. You have an amazing story. And I see you as someone that has done a lot of work understanding the mechanisms of bioweapons and how to heal. And there appears to be many similarities between COVID and Gulf War. And I thought we could start by telling you how you came into this particular space, as we said before, a matter of survival when we started this podcast.
Well, I have a number of members of my family who are veterans. And so naturally, it's something that is close to my heart to try and help them whenever they need assistance. So my stepdaughter served in the Gulf War, and she was a crew chief on a Black Hawk helicopter, and came back and came down with a number of symptoms, along with over 100,000 other veterans of the Gulf War, including Australian, British, Canadian, and a number of other countries supplied forces to the Gulf War. So this is not something that was strictly limited to the American service personnel that served there. So we knew that this was going to be complex because when we started looking into this, and I had to do this on the side because it wasn't part of my normal duties. But since I was an endowed professor and had my own funds under my chair, I was able to slip some of this research in. And we were able to look at the problem of Gulf War illnesses as a composite problem. It wasn't just one thing that was causing the problem. They were both chemical exposures and biologic exposures. And for a few cases, there were radiologic exposures, mostly from depleted uranium munitions, but that turned out to be very minor compared to the other two. And they were kind of split about half and half. That we found that many of the veterans were exposed to chemicals, a number of different chemicals. Of course, there were about 150 oil well fires that were on fire at the end of the Gulf War. There were also contaminants that they were exposed to, including a few to biological and chemical weapons. So that was a possibility as well. And then we had a problem with the vaccines that most of the forces were given. And some of those vaccines are problematic that they were given because they were essentially experimental. And that's something that was a very difficult problem because we later found out that some of these vaccines were being tested in the prison system in Texas. And so this added another layer of complication, but it allowed us in a way to figure out the background on how these ended up in Gulf War veterans. So let's kind of break this down into two major classes of exposures, biological and chemical. So we've worked on both of these. And it turns out my topic today that I'm going to be discussing, membrane lipid replacement, is actually used in the therapy for both of these exposures. So I'm not going to really cover the biologic exposures. I've talked about this quite a bit online, and my late wife and I wrote a book on this called Project Daylily, which we had to change all the names and places and institutions and so on and so forth so that we couldn't be sued to bury the book. So it's based on a true story, but the names are fictional, the places are fictional, the institutions are fictional, but everything else is absolutely true. So that's something you learn you have to do to protect yourself legally.
Yep.
So that covers really talks mostly about the biologics because that's the one that had the most intrigue associated with it. But the chemical ones were a little more straightforward than that. The chemical exposures, more recently, I've worked on that. We published a few papers now on how to treat these chemical exposures, and we've had a lot of good results with the veterans. And it turns out to be very useful for a number of other exposures from Lyme disease and other types of diseases which result in, for example, mold, other things where there are natural toxins involved. And those toxins, like the chemicals from the Gulf War, are very hydrophobic. That is, they tend to seek places in your tissues and cells that water can't penetrate into. So it's in the non-water, more lipophilic parts of cells. And of course, membranes, which I worked on for the last 50, 60 years, are an important part of the sink that those chemicals find their way into. And when they do that, and when they get in these areas of the cell, they cause a lot of problems because there are a lot of very important enzymatic information, signaling, control, regulation, nervous system activity that goes on because of the membrane functions, and you have to maintain those membrane functions. The membranes are also important because they form important barrier functions, not only separating cells from one another, but there are also other barriers that contain membranes that separate the cells from the tissues, the circulatory system, for example, the gut system, the brain, and other places around the body where these barrier functions are extremely important. They keep out pathogens, they keep out toxins, they keep out chemicals, and so on. So when they're damaged and become leaky, all these dangerous things can enter into these compartments, like the brain, for example, or various tissues, various organs, and so on, and can cause a lot of problems because of they just tend to accumulate and damage the membranes and other sensitive enzymatic machinery and even regulatory machinery inside cells. So it's something that we've spent a lot of time trying to correct.
It's almost as though you could give a model for disease as a leaky membranes, or leaky. Like when you think of a leaky gut, you've got a leaky blood-brain barrier connected to neurological disease. That once you're strengthening those barriers of the cell membrane, it fixes things.
Well, this is
kind of
how
a simplistic idea I'm trying to sort of break down
this,
for the audience.
well, you can think of it this way. This is how people get sick from microorganisms, because whether they're inhaled, and we're talking about the epithelial barrier in the lungs, or they get consumed, and we talk about the epithelial barrier in the gut, or the skin. How are they absorbed into the body? This is how they get in, because the barrier is not functioning properly, and it's permitting the entry of these pathogens into our system. So barrier function is incredibly important. That depends a lot on membranes, and the various membranes that help maintain that barrier. And of course, I've worked on membranes my entire career, so I know a little bit about this. And I know a little bit about the barrier function of membranes and how they can be preserved, and when they're broken down or damaged, how they can be repaired. And so this really gets into my main topic today, which is membrane lipid replacement, because we know that there are various membranes in the cell that perform all sorts of different functions. When I first got started on this, I was very interested in mitochondria, because these are the little organelles inside every cell of our body that are kind of like the little batteries for our cell. They provide the energy for everything that goes on in the cell. So you can imagine every function of a cell requires energy to perform all these different functions, whether it's moving molecules around the cell, or it's providing energy to make new molecules, or whether it's providing energy to maintain the barrier function and to separate various compartments within tissues and within cells. All of this turns out to be very important. And when these membranes break down and the barriers break down, then things start to fall apart in cells, and this can produce what we call disease. So it's not only important in terms of disease, but it's also important in natural aging, because as we age, these barriers tend to become dysfunctional and tend to break down. And so people are more susceptible to infections, for example. Their immune systems don't work as well. All the sorts of things that go on that are necessary to maintain normal housekeeping functions in cells tend to start to wind down, and then things start to go wrong. And so ultimately, this can result in aging as things start to fall apart. So these barriers are incredibly important, and we have to maintain these. And the most basic of the barriers is the membrane of the cell. And of course, there are all sorts of membranes. There's a plasma membrane that surrounds the cell, but inside the cell, there are all sorts of other membranes that provide unique specialized compartments. So we have the nucleus, for example, that contains a genetic material, and we have all the machinery that's necessary for cellular division and maintenance and gene expression and so on and so forth in the nucleus. And then outside the nucleus, all that information has to get expressed. So whether we're making new molecules, which happens in another membrane system called the endoplasmic reticulum, All of that occurs on membranes. And in, I mentioned the mitochondria, which has a dual membrane system. There's a membrane that's outside the mitochondria, which provides a shell. Inside that shell, there's folded membranes, which are called the inner membranes of the mitochondria. And that's where the action occurs of making high energy molecules like ATP. And there are also other functions that occur. Mitochondria are very important because they provide a number of functions. They provide messages that are used both within cells and cell-to-cell communication. They provide regulation for very important and critical ions like calcium ions. They provide unique molecules like steroids, for example, which are necessary for all these communication systems that we have. They provide synthesis of various lipid types of molecules, which are very important. So they're incredibly important. They also provide a source of innate immunity, which is a very important form of a very primitive type of immunity in our systems that we find are very necessary for responses against very primitive types of structures that occur both in pathogenic microorganisms, but also in allergic molecules and allergens and so on, and various other things. So this multiple membrane system that we have in our bodies, in fact, every living system has them, are incredibly important for barrier function, for enzymatic machinery that provides new molecules, for synthesis of proteins and carbohydrates and other things that go on, and synthesis of fats as well, and communication. So we can think of, at the gross level, communications in our nervous system, for example, depend upon membranes, because for communication through nerves to occur, there has to be depolarization of the membranes. And here's another area where membranes are so important in providing a barrier for electricity inside our cells. So we know that there are these electricity barriers as well, and they're maintained by the same membranes, and there are various pumps that pump ions across the membrane to maintain this electricity barrier. And it really forms the basis of nerve transmission. So everything that we see, do, feel, touch, smell, taste, whatever, all occurs because of membranes and membrane barriers, which need to function to provide the messages that tell our brains that we're in a certain environment, that we see certain things, we smell certain things, we feel certain things. We have tactile responses, we have other types of responses. And of course, when there are noxious materials around, which can be chemicals, heat, cold, whatever, we feel these, we sense these as well. And so we have specialized nerves that communicate these, and all that depends upon membranes. Not only the membranes themselves, but the barrier functions that help maintain those membranes and the energy systems which occur. And when some of these things go wrong, then all kinds of different symptoms occur in our systems, in our cells, and so on. And some of the basic symptoms that can occur because we don't make enough high energy molecules. And so one of the things that happens in almost all types of illnesses is that people suffer from fatigue. So fatigue is one of these universal symptoms that occurs with practically any type of illness, whether it's caused by infections or chemicals or allergens or whatever. Fatigue is one of the side effects of it, and that's because of our mitochondrial function, which is not obliterated, but it's not working at its peak performance. And so all of this depends upon these inner mitochondrial membranes, which provide the energy for the cells, so that this is now perceived at the organ and organismic level as a fatigue. So loss of organ function or a fatigue feeling, which is really loss of the ability to perform certain functions without being exhausted or not replenished by sleep and so on. And if this occurs for more than 6 months or so, we call it chronic fatigue. And chronic fatigue is one of the most common symptoms of all types of illnesses. It occurs in almost every type of illness that you could imagine. You suffer from chronic fatigue because of it. So we're talking about most infections, for example. We're talking about most conditions which result in impairment of organ function. All of this is really resolves around the mechanisms of fatigue and loss of mitochondrial function. And of course, loss of mitochondrial function helps you from healing as well. So
Yes.
it prevents you from recovering from disease. And then sometimes our functions, loss of those functions go on long after the disease is gone. There's no evidence, for example, for a pathogen, but we still have symptoms that linger. And sometimes this is called a latent disease or reoccurrence disease, but it's really lingering effects because of the functions that haven't been maintained. So something that I've worked on recently, long COVID, which is a condition where you have the symptoms of COVID that last long after there's any detection of the virus in your system. So these are things that occur that need to be fixed, more or less,
Mm.
and very little is known about them. But so one of the things that happens with these lingering, persistent portions of disease is our barrier functions are not being maintained properly. So we're not making the proper energy molecule. We're not maintaining our function in terms of our nervous system. We're not maintaining our gut function and so on. And so these are the things that happen with disease that we're trying to do something about. And so for all of these situations, membrane lipid replacement is important. Why is it important? Well, the matrix of all of our membranes is made up of a lipid bilayer. And this has been known now for over 100 years that membranes do contain a bilayer. We didn't know how it was organized beyond that, but that's where my work with Professor John Singer, who unfortunately passed away some time ago, what we found was that the lipid system forms the matrix, but it forms a fluid matrix, at least in certain areas of the membrane, so that things in the membrane are capable of moving around. It turns out this is very important for cell signaling. It's very important for cell communication. It's very important for many of the functions of cells. It's important for messaging and new messages from outside the cell to get transmitted across the membrane inside the cell. It's important for capture of various metabolic and nutrients in the system and transporting them into cells. It's very important for getting rid of the waste molecules from cells that have to be transported out and sent to our gastrointestinal system or urinary tract system for elimination. So all of these things are incredibly important, depend upon the enzymes and the structure of membranes. And so that's something that I've worked on my entire career. And matter of fact, it's a few years ago was the 50th year of the fluid mosaic membrane, which is a textbook model of membranes. And so I produced a number of papers on the 50-year anniversary of that
Mm.
membrane with Professor Singer. That I was a graduate student with Professor Singer, so this was actually my graduate thesis work. And so it's very rewarding to me to see how many different areas of medicine and biology have used this concept to help explain many of the basic things that go on in life, but also many of the things that go wrong in life,
Yeah.
including illnesses, various illnesses and infectious diseases and a variety of other things that can go wrong, depend upon the maintenance of membrane structure and in turn barrier function of within the cell and within the organisms that we are. So it's important for practically everything we do. As I mentioned, everything we see, smell, taste, feel, and so on depends upon our membranes and our cells that process this information in our brain and so on. And so when this multi-structural arrangement called membranes goes bad or gets damaged, we have to repair it. And so we've evolved with really very intriguing sets of processes to repair, rejuvenate membrane systems so we can get back to where we were in terms of our health. And we've developed a very natural way to help with this repair process, because one of the basic things that happens is the lipid molecules in membranes get damaged. Mostly this happens because of oxidative free radical damage, which occurs during every chronic and acute illness, free radicals get produced, and these damage not only the membrane, but they can damage our genetic material as well, and damage various organs like the mitochondria and other basic organelles inside cells that are absolutely essential for their function to maintain normal cell physiology. These get damaged, and of course, a lot of this communication occurs through ion channels and membranes, and ion channels turned out to be very sensitive to the lipid molecules that they bind, and they have very specialized binding sites on those ion channels that bind very specific lipids. And when this gets damaged, they don't function properly, and this can result in leakiness of the membrane. The ions in the membrane pass through the membrane, become leaky, and change the electrical properties of the membrane. And it also happens that we can't take in the proper materials as well because of it. So all of this goes back to kind of what happens in the matrix of the membrane, which is the lipid part, and in ion channels, for example, are proteins that are intercalated into that lipid matrix and span the lipid bilayer of the matrix and form the pores that we can have ion communication coming in, going out of cells at different ion channels, for example. And then we have different parts of the membrane that can form into different shapes, and some are invaginated and form vesicles and bring things in from outside the cell. Some things fuse, vesicles fuse with membranes and expel certain materials outside the cell. And then we have contact points on membranes, and some of these are mechanical. For example, we have cells that come up against other cells, and they're kind of bound together because of different forces that attach between the two membranes of the cell. And some of these form actual junctions of the cell. Some of those junctions actually have holes in the junctions, or for ions to pass between cells and messages to pass between cells. Others form attachment points where extracellular matrix molecules can attach to them, or the cells can attach to other things and which are necessary in forming the structure. So the structure of an organ is totally dependent upon not only the barrier function of the membranes of cells, but on these extracellular attachments that form with the extracellular matrix and with other cells and other systems to form blood vessels, for example, or extracellular matrix that are important, everything from cartilage to bone and other things are important to form the structural aspects of complex organisms like ourselves.
And it's
All
interesting
this
that
depends upon membranes.
I mean, it's interesting that when you're talking about chronic fatigue, when someone is so sick and the energy level is so low, what's the first thing what can they do first to restore cell membrane? Are you saying that you would put them on NT factor? Is that something that's given?
Yeah, let me
by
get to that
drip
because
or
I'm working up to that.
Yes, yes.
I promise you, I'm working up to that. So we get back to the basic membrane, the basic fluid mosaic membrane, where there's a fluid matrix. At least part of the membranes are fluid. And what makes that fluid are the lipids. So the lipids are incredibly important in the membrane. And the lipids have a certain turnover rate. We have to replace them because they're very sensitive to damage. They're very sensitive, for example, to damage by free radical oxidants, which are produced naturally, and we get many of these. They're very sensitive to damage by chemicals. Let me get my earpiece back in here. So we need to have a source of this. So a good source, for example, are lipids that we eat, we ingest in, but a lot of those lipids are damaged before we even get to them. As a matter of fact, a lot of the lipids that we eat are damaged already. So that's like the old story of computers, junk in, junk out. If you take in lipids which are already damaged, they're not going to help you. We have to take in pristine lipids.
But even like organic food? Are you saying that even organically grown fresh food
even organic food. The problem with non-organic food is there are a lot of chemicals and other things in them which really interfere with a lot of different processes, and they could damage your system independently. But even if you take organically grown food, if the lipids are damaged, if they become what we call rancid or oxidative damage to those lipids, they're not going to help you, because a lot of the lipids that are important in membranes are lipids with double bonds in them. And those double bonds are very sensitive to oxidative damage. So we have to repair those kinds of lipids all the time. So we're always repairing them night and day, 24 hours a day. We're repairing these lipids. We're bringing in lipids from outside and repairing them because a lot of this can't be done enzymatically because it's just too difficult to replace these double bonds in lipid molecules. So we have to bring in, it's much easier to bring in new lipids that contain these. So we need a pristine source of these lipids. So if we go to fresh foods, for example, we can get more of these. If we go to certain types of food, we can get more, but sometimes we can't get as much as we need, even from food. So we have to have supplement. And to supplement, we need a lipid that is absolutely pristine and is protected from oxidation so that it doesn't get oxidized during storage or goes rancid. And then it's not very useful to bring in things that are already oxidatively damaged. So the important thing about membrane lipid replacement, which is what I'm going to really talk about today, is that we need protective molecules to prevent this from happening before we ingest them and after we ingest them and when they actually get in our cells. And so thankfully, there's been a long evolution of protective molecules that goes back to the earliest form of plant life in our planet, and probably other planets as well, as a matter of fact, because a lot of this may have come eventually originally from other planets. But these early life forms, for example, developed membranes as part of the area to separate off and form a unique cell. And they had to have this capability of fluidity of the membranes and other things to function just like we do. So they needed particular types of lipids to form those membranes. And if they didn't have those lipids, they had to get them somehow. So they had to be around in the environment. So plants, for example, have evolved with molecules that protect lipids from getting oxidatively damaged. Now, some of these molecules are called inulins. They're really fructooligosaccharides. So all plants have these inulins, various families of them, and they're always used as protective molecules for the lipids in membranes. And without these, for example, plants couldn't survive. So the plants in the far north that are frozen half the year, they wouldn't survive, they'd be dead. So the inulins protect against freezing, protect against temperature changes, they protect against oxidative damage, they protect against enzymatic damage, and so on. So a lot of these things that we take for granted are really basic to some of the things that we do. So we've developed over the last 20, 25 years or so a way to protect the membranes using a natural lipid supplement. And it's was called, I didn't have anything to do with the name, by the way, it's a commercial name, NT-factor lipids. So these interfactor lipids provide the basic building block for what I did design myself, the word or the term now, because it's a registered term, membrane lipid replacement. So this is how we can replace the lipids that are the most sensitive to oxidative damage. So we have a source, a plant source, for example. We can get rid of the lipids that we don't want and get the lipids we do want, these highly double bonded lipids that contain the really most important types of lipids that we need in our brain, for example, in our membranes and all our cells. And we burn these up at a high rate, so we need to replace them. So one of the things that happens when you age, as well as happens when you have disease, is these get damaged, and they have to be replaced. And so we have to have a way to replace them. Excuse me, my computer's trying to fall off the table here. So a little minor adjustment. Okay, so this is what the natural replacement product, membrane lipid replacement, does. It replaces these. So we've evolved from the hunter-gatherer stages where we needed to get these lipids because a good source were animal tissues, for example, that have a high amount, just like we have of these membrane lipids. But we didn't have this source very often. We only had this source once a week, once every 2 weeks, or whatever. So these molecules were really important. So we've evolved with a system to not only grab these molecules when we eat them in our small intestines, but not only to bring them into our body and store them as quickly as we could and send them around our body. So we've evolved with this really complex system to bring these in, to store them in various ways and cycle them around, bring them through the liver, make new molecules out of them, which are more human molecules, and then send them off to all the different places where they're needed. So this is why membrane lipid replacement is so important. And as I mentioned, in disease and aging and so on, this function tends to go down. So we need these molecules more than ever, and we need membrane lipid replacement more than ever as we age or as we're exposed to disease processes and so on. So this is where this commercial product comes in. It supplies these in a way that are useful, in a way that won't contaminate you, so it won't give bad molecules into your body. These are all good molecules, and they're protected. That's the patent protected as well, by the way.
So it gets through the stomach acids through to the body.
Oh
And
yeah, that protects against that, protects against acidity, and protects against oxidation, enzymatic damage, and so on. These inulins are so important to the survival of these. And of course, if you take a plant source, well, what happens if you eat plants, for example? And we get them in small quantities in plants. Those molecules are already protected with the inulins because the plants needed them to protect them. So you'll get a source of inulins in with that. But it's very important to have a little excess of these inulins to help protect the materials that we eat or that we pick up in a number of different ways. So that's why we need the inulins along with the lipids. We have to have both of them. And not all the lipid products have this type of protection. So the lipids go bad very quickly. They get oxidized very quickly. They get damaged very quickly. They're very sensitive to temperature. So they're hard to store. So this is why we have to be very careful with this material that we bring in.
The only way to take this product is orally. You haven't, there's no experimentation with using it by IV or any other method.
Well, it turns out these molecules are so important that we have evolved with the method to our oral digestive system is all you need.
Okay.
You don't need to put these in intravenously, although in acute cases, for example, in Eastern Europe, this is very common for acute hepatic toxicity, for example, for poisoning and so they do use IV lipids of the same type. But turns out to be much more used in Eastern Europe than we use it. But again, it turns out to be very important in emergency care. For example, if someone has a heart attack, for example, one of the things that happens are these lipids get damaged right away because there's a burst of oxidative free radicals when you have a heart attack. And that damages all the lipid membranes and so on and causes ischemia and other things. So you can't generate the energy in your cells. And this causes all kinds of problems with your heart muscles start to die and so on. So one of the things they do is they give IV boluses of these lipids along with oxygen and other things to help restore the membranes and start the healing process after damage has occurred because, for example, of a heart attack and ischemia that happens because of that. So very important. So now back to what really goes on when you bring these in and why so many of our functions are dependent upon the activities or the stability of our membranes. So we're replacing these lipids all the time anyway. So this is a major thing that goes on in your body all the time, that you're bringing lipids in through the foods that you eat. You're modifying them to make them more human in terms of their structures in your liver and other cells, and you're sending them all over the body, and they're picked up, they're transported in, they fit into various membranes, and older structures are pulled out, and they're sent. Eventually, the stuff that's damaged is pulled out, sent back to the same mechanism, but it ends up in your GI system as trash, and it goes out in your stool. So you eliminate the bad stuff as well as bringing in the good stuff. And all of this is important because if you don't eliminate it and the damage piles up in your system, that's also bad because that could poison you and cause other problems as well. So we have to have all these systems working in concert to produce a desired effect. And it's very important in healing after damage that's occurred, damage to infection, damage to physical damage, for example, or other types of damage. We're even working on various types of damage caused by electrical potential, for example, damage from some people are very, very sensitive to Wi-Fi and transmission towers and all kinds of electrical impulses that they receive that could damage their body. And so we're actually using it to help repair that as well. So it turns out it's incredibly important for all sorts of things. So as we take this in, we can supply excess in the form of the NT-factor lipids, for example, which is taken in orally, and your body processes it very quickly and sends it around to our body and starts to rebuild the plasma membrane, starts to rebuild the mitochondrial membrane so that we have more energy. And then once we get the energy, then all the systems can repair. And so there's less fatigue, there's less problems. All the sensory systems get to be repaired. Our skeletal muscle systems are repaired and so on. So we slowly recover from a disease process or from trauma, from injury, from all these different things that can happen to us. So this is very important. So going back to my experience from the Gulf War, we've used the NT-factor lipids to provide a number of different capabilities of our injured personnel. So I'm going to talk about chemical injury near the end of this talk, but let's talk about biologic injury and injury from EMF and so on. This is so important that we have these membrane lipid replacement molecules there, not only because we have to repair the system that's damaged, but we have to repair the barrier function of our systems. And these are things that break down because of chemicals, break down because of electromagnetic forces that are depending on us and so on. So we have to maintain these barriers and we have to build them back up. And that's what the membrane lipid replacement does, builds up the barriers. And in the mitochondria inside our cells, one of the most important things that it does is it returns the energy functions, the synthesis of high energy molecules to the mitochondria. So we've seen, for example, anywhere from 20 to 45% improvement of mitochondrial function when people take the NT-factor lipids. And so when we first started working on fatiguing illnesses, so these are things like, of course, Gulf War illness falls in the category of fatiguing illness, but also chronic fatigue syndrome and all kinds of other illnesses. Fatigue is the most common complaint of all patients seeking medical care. So it occurs in virtually every type of illness or exposure or damage that you can imagine. So the number one thing that we need to do is repair mitochondria. One of the first things we need to do. What is it in a heart attack that has to be repaired first? The mitochondria have to be repaired first. What happens in infections? The mitochondria have to be repaired. And what happens in all these other injuries and everything else that goes on? The mitochondria have to be repaired. So that's one of the primary functions of membrane lipid replacement, is to repair the mitochondria so we can return that intermitochondrial membrane to function. And when it's functioning properly, there's a transmembrane potential across the intermitochondrial membrane of about minus 150 millivolts. So mitochondria are little electrical capacities in themselves. And if that minus 150 millivolt falls, then we can't make the ATP. We can't make the high energy molecule. It's also important in nerve impulses. If the resting potential of the nerve across the plasma membrane, if that potential starts to fall, we can't have nerve transmission. So it damages the nerves. And so all kinds of things depend upon the electrical properties of cells, and membrane lipid replacement helps maintain those electrical properties, which are absolutely essential for a number of different functions. So daily I take, and a lot of other people I know take, the membrane lipid replacement. NT-factor lipids, for example, is one product. And this helps repair the mitochondria and return to function and slowly repair our bodies, slowly return our normal function. And as a part of this, it also helps repair our systems, which remove our damaged molecules from our cells and shunt them outside our cells, package them up for delivery to our gastrointestinal system and our elimination in the stool. And so, for example, damaged lipid molecules, this is how they find their way out of our system. We can't just keep accumulating these things. We blow up bigger and bigger with all these damaged molecules. We got to get rid of these. So to maintain the systems, we have to get rid of the damaged molecules. If we can't repair them, we've got to eliminate them. So using kind of that as an idea, and the fact that a lot of the damaging molecules, particularly from petrochemicals, for example, which is one thing I worked on from the Gulf War, a lot of the veterans were damaged because of petrochemical exposure, from burning oil fields, from fuel direct contact, from burn pits, from all these different sources of petrochemicals, burn petrochemicals that were just in the environment, so exposure, skin exposure, breathing it in your eyes, in your everything, just everywhere.
Yep.
So these patients had a lot of problems, and it's very hard, it turns out, to get rid of these chemicals once they get in your system because they're very hydrophobic and they partition, because it's a
Ah,
chemical property, into all the membranes of your cell and also all the little lipid droplets and globules. There's one thing I didn't mention is that when our lipids come into our cells, there are all kinds of different compartments where we store these lipids because they're so important. Some are stored in lipid droplets, for example, which are a specialized lipid thing, and that's where we store fats, for example. But there are other, what I call lipid globules, which are kind of smaller than lipid droplets, but they soak up the lipids and store them, and they're shunted around the various places around. There's all this activity in cells where they're going from mitochondria to the endoplasmic reticulum, back and forth to other things, vascular to the nucleus, to the all kinds of things going on. So all these lipids are being shunted in these little uh, tiny little lipid globules, and so very important in maintaining the homeostasis of our cells. So I thought, well, if all this is going on, we ought to be able to use these lipids to slowly encapsulate these damaging petrochemical molecules that are brought into our cells naturally and tend to accumulate and cause a lot of damage, because a lot of these things can cause chemical poisoning. And when
yeah.
they build up in the cell and we want to get rid of them, of course, one of the things people try to do is they have a very rapid detoxification. That's the worst thing in the world you could do, is rapidly detoxify. It lets all these things out and they do the damage. And so what you want to do is slowly remove them. And this is what
Mhm.
membrane lipid replacement does. It gets around and surrounds these hydrophobic molecules with the natural lipids, and then it shunts them into these little lipid globules and membranes and back and forth again over and over until eventually they find their way into what we call exosomes, the little lipid bilayer formed vesicles that eventually use to take out the trash of our cells.
Yeah.
These fuse with the plasma membrane, they're expelled. So they get out in this, and then they're picked up by other scavenging cells that circulate around, the white blood cells, for example, and they're picked up by other systems. And also we have lipoproteins and other things that are circulating around and pick up these lipids. So we have various ways of moving these things around that we've evolved with over the years. Our cells have evolved with all this machinery for just dealing with moving the lipids around. So I reasoned that, we could use this maybe to get rid of the hydrophobic lipids in cells, but we don't want to do it too quickly, because if we do it too quickly, we could actually poison people. And so
So
we
are we
want to do
taking
it
lower
slowly.
doses at the start, or this you can't
No,
overdose?
it turns out it doesn't really matter. These things are so important that we have a self-corrective process. If we bring in huge, massive quantities of it, we store it. If we bring in low
Ah, okay.
quantities of it, we concentrate it. So we've evolved with all these systems because we to try to maintain the proper equilibrium. So it turns out it doesn't really matter. People ask me, well, should I take my lipids all at once or space them out, take a little bit in the morning, a little bit in the evening, or whatever? Turns out it doesn't really matter, because we've had these systems where if we take in a lot at once, we expand the system to deal with that. If we take it a little at a time, we developed a system to deal with that too. So
Mm.
we have a marvelous capacity to maintain and grab these things. The important thing to know is the important molecules that we take in, virtually 100% of these are taken in, are transported down to the liver first, because they come in generally through the hepatic duct down to the liver, and then they're in the liver. The molecules need to be humanized, because let's say we eat plant molecules or other animal molecules and so on. Well, the human makeup of these molecules is a little bit different. Not some of the molecules are very similar, but other molecules are a little different. And we have to humanize them. In other words, we have to enzymatically convert them to more human form. And then a lot of that happens, not all of it, a lot of it happens in the liver, which is why the liver organ is so damn important. Because you need to, before you send these molecules around your system to be picked up by all these different types of cells, we have to make sure that they're human molecules. They have the structures that we need to support human cells and human physiology. So that goes on mostly in the liver. So the liver is an incredibly important organ, and that's where these molecules first come in. So again, all these things go on. They're going on all the time. But just to let you know that that does work. If we find out something on how a system works in our body, we can use that as a natural way to heal your body. And that's essentially what we do. We use this natural healing process of our body, and we just feed it a little bit more, a little bit more, a little bit more of these natural molecules that we need to heal. And so our body does the rest. We don't have to do anything more. All we have to do is supply it. And so there's no magic drugs or magic things that go on. It just all uses the natural systems that are already there. So that's the marvelous thing about membrane lipid replacement. We don't have to do anything special. We just have to supply the important molecules and let our bodies, do our thing. Let the
But of
cells
course,
do their thing.
it's obviously good and important to eat well as well, and not to
Oh,
eat
yes.
junk food.
Well, of course, again, I mean, I'm talking about lipids, but there are a lot of other things that are necessary as well. And we have things called vitamins, for example. We have things called minerals. And all these things are important because they all work together and provide the necessary functions. So we can't forget about those. I'm just talking today about liver.
Yeah. So it's like jump starting
Membrane
the
lipid.
body.
Yeah, so I'm
You're
talking about the
essentially
role
jump
of
starting the body
lipids.
to work properly, because when you're
Yeah.
sick, your energy goes down. So you're like a car that's not running on all
Well, the first
cylinders.
thing it turns out we have to do when somebody's sick or injured is that we have to start the healing process. To start the healing process requires energy, requires high energy molecules in the cell. To rebuild any structure in the cell, we need energy. To perform any function in the cell, we need energy. To eliminate any dangerous molecules or damaged molecules, we need energy. So energy is required for everything that we do. It's required for gene expression, to make new molecules, to get rid of old molecules, to repair damage. All that requires energy, and 90% of that comes from your mitochondria. So we need to make sure the mitochondria are repaired first. So that's why we've concentrated on membrane lipid replacement to repair the inner membrane of the mitochondria so we can maintain that minus 150 millivolt electrical potential across the membrane, because this is what drives the production of high energy molecules. And people talk about the electron transport chain and all the ways in which we have to transfer electrons and protons and so on. All of that depends upon the membrane, because you can't have that unless the membrane is properly composed and it has the proper matrix, because the membranes themselves can become leaky. And so we can have molecules leak across the membrane unless they're tightly together. And this all happens because of an effect called the hydrophobic effect, which is a water effect which drives things like lipids, which are bimolecular structures. They have a hydrophilic head group, and they have these long hydrophilic acyl chains that come off of them. So they all line up together on one side, and they also line up on the other side to form a bilayer. And that's the basic structure of a membrane. And then intercalated into that are various proteins and other structures and so on. So that's the essential starting point of a cell. That's how cells evolved in the first place. Without a membrane, there is no cell. Without a membrane, cells never would have been.
And it's not just theory here. You've done a lot of clinical trials. Like, if we wanted to talk
Well,
about some of the things that
that's how we have to prove that what we're talking about really does help you, is to perform a clinical trial.
but
And
not all
so,
supplements do that, though.
well, no, a lot of the supplements that you see or buy in the store, they depend upon some of the basic uh, research that's gone on over the years on basic requirements, metabolic requirements, basic nutritional requirements, and so on. They depend upon that information. But if you come up with something a little bit different, you've got to do clinical trials, because you have to go all over again and reprove it, reprove that it really works, and it works over and over again, and it repetitively works. So that repetition is very important. So this is why we have to run a clinical study. So we've run a number of clinical studies with membrane lipid replacement, and where we started really was mitochondrial function, and we were able to show that if patients in a clinical trial take a membrane lipid replacement lipid supplement, that their mitochondria are repaired. And we can show that because we can take a sample of cells from their body. In this
All
case,
right.
we took blood. We took the white blood cells from their blood because they have mitochondria. The red blood cells don't have mitochondria. So we have to take the white blood cells from their blood and look at their mitochondria. And we can see that function of the mitochondria by using very specific dyes that are activated by a laser, and those dyes measure the electrical potential across the inner mitochondrial membrane. And we know when it's minus 150 millivolts that they're functional. And there's all kinds of experiments that are done to prove that, including the actual measurement of ATP production. But the easiest thing to do now that we know that these dyes work is to put in the dye, and the dye goes into the inner mitochondrial membrane, and we can read that dye with a fluorescent microscope. And so we can look at a cell, for example, under fluorescent microscopy and put in a specific dye called rhodamine or JC1 or any number of different dyes, and those cells will show fluorescence in the mitochondria. And you can see the little mitochondria either around the nuclei or around the periphery, depending on the cell type, and they're all lining up properly, and we know that they're
That's
all
amazing.
working like they should. So we
That
can
must have
see
been very
that.
exciting to be able to produce a product, see it in
Well,
patients,
that's the other
and
thing that's
actually not just see them recover, but also underneath the microscope to go, wow, the blood actually has changed.
well, what's I think the most exciting about this is to see how useful a simple nutrient like this is, because really anti-factor is really a nutrient. It's a food. It's not a it's not a drug. It's a
Mm,
food. So it's something that we bring in every day. We just don't bring it in the right quantities, and there's some structural things that are not right in the stuff that we bring in, so we have to change it around. So the anti-factor lipids are the stuff that's easy to change, and it's not damaged oxidatively. So free radicals have not damaged it before we get it in, and then it can build the new membranes, build other structures in the cell, and bring the performance of ATP production up to peak performance. So cells were humming, they're operating just like they should. So all the functions of the cell operate. So the repair functions in our tissues, and when they get damaged, we need to have that energy to help that repair process. So when you get a disease, there's damage that occurs. That's why you have a disease. There's damage that can't be repaired naturally. And so this is what performs a natural function. So when we have other types of, let's talk about infectious disease. You think, well, what about infections? Or they help with this? Well, infections, it's not so important how they're helped. It's more important, can we limit the damage that they do? And can we
mm,
actually eliminate them? Can we either stop them from dividing or actually kill them? And that's using various types of anti-metabolic drugs or cytotoxic drugs to either kill them or damage them. So yes, even membrane lipid replacement, we use it, for example, right after cancer chemotherapy to help repair the damage that's done to normal tissues by the cancer drugs. And very amazingly, there's a reduction in all the bad symptoms that occur because of the cytotoxic therapy. We can eliminate a lot of this, and people recover much quicker from the therapy and can go on to lead normal lives. They doesn't linger around. Same thing after disease processes, for example, like COVID. A lot of people have had trouble with long COVID, the lingering effects long after the virus is gone of the damage that's occurred and accumulated. And how do we repair that damage and bring them back to normal situation? That's the importance of membrane lipid replacement. It stops the autoimmune reactivity. It stops the leakiness of the membrane. It stops all of the damage systems that go on, and the damage that tries to repair itself but doesn't quite do it correctly, that happens because of things like viral infections or bacterial infections or other types of protozoan infections and so on.
So,
All these cause damage.
so instead of thinking of a disease as attacking, say, the toxins, you're talking about in terms of using nutrition to almost push out the bad stuff. So with
Well, we've got to get rid of the bad stuff, and that's the way to do it. So, what I'm trying to say is it does not interfere with the actual treatment. So if you use an antimetabolic, for example, or a cytotoxic drug to kill an infectious agent, or interfere with its function, like we do with viruses, or we might want to kill a protozoan or a bacteria that we find in Lyme disease, let's say. So we need very specific drugs to do that. But it takes people a long time to heal, even after we've knocked out the microorganism, because this damage that occurs accumulates and goes on to in itself cause further damage. So we need to repair that. That's where the anti-factor lipids come in. So
with
reduces the symptoms of disease and reduces the symptoms of the aftermath of the disease.
so with the Gulf War syndrome, you found that there was a lot of mycoplasmas in people's blood.
So that's,
so
as I
when
mentioned before, people were damaged, a lot of them because of contamination of the military vaccines that they received, but they also may have been exposed environmentally. It's less a problem with the infections than the chemicals. The chemicals were all environmentally obtained,
So-
but a lot of the infections were not. They were obtained from the contaminated vaccines. That's why, for example, the French forces that fought in the Gulf War didn't really have any incidents of Gulf War illness. They didn't take military vaccines. They used prophylactic antibiotics to fight off infection. But unfortunately, the Anglos, the British, Canadian, Australian, American forces used vaccines. That was our downfall. That's why almost all the Gulf War illness cases were from those nationalities. So, so what we found was we had to do both. If somebody was exposed biologically, we had to figure that what that was, and we did find a particular microorganism that was probably the most dangerous because it led to lethal forms of the disease and probably accounted for more than 90% of the lethal incidence of the biological exposures. Turned out to be a mycoplasma, which is a very, very primitive bacteria that is everywhere. There are all kinds of mycoplasmas. You find them in plants, for example. You find them all over. So they're everywhere. But there was a particular type of mycoplasma that turned out to be very pathogenic, caused a lot of damage in humans. And so this particular one that we found turned out to be extremely unusual. In fact, it was so unusual that it wasn't heard of until a few years before the Gulf War, and it turned out the U.S. Army was studying this at the Armed Forces Institute of Pathology in Washington, D.C., and they actually patented this particular, not really the microorganisms, because you can't really patent that, but they patented the process for detecting it. So they actually processed the ways in which you test and detect mycoplasma. So we were looking at the Gulf War veterans after the war. We knew they had chemical exposures. We knew they had biologic exposures. And what attracted me to work on first, that we first worked on before the chemicals, we worked on the biologicals. The reason we did that is that I had a lot of families that I was working with because at the time I contacted people saying, you know, my family got sick after I returned from the Gulf War. They're showing some of the same symptoms that I have as a Gulf War veteran. And so we knew that this had to be a transmittable disease and an infectious process that we're dealing with in those particular instances. So we'll separate off the chemical exposures at the moment. So let's say half were caused by the biologic. Turns out the people that had biologic exposures also had chemical exposures, which made the biologic exposures even more damaging. But their family members are getting it. So that's the first thing that we studied were the families, the spouses and the children becoming autistic, for example, which led me into autism spectrum disorders and the role that some of these similar, not the same, but similar types of microorganisms might cause in children, along with chemical damage that we know occurs in children. So you can think of autistic children in a way as kind of in a small scale what happened to the Gulf War veterans, because some of the same problems occur in the veterans that we see in children. So working with the families allowed me to actually prove that that this was part of these veterans had an infectious process that spread to families because we found the same unusual infection in the family members. So when we treated them, and eventually many of them recovered, and what we found was that we could no longer detect those infections in the
Mm.
families. So we had to conclude that this infection was very important in the
So when you're talking
process of
about
their disease.
treating them, you used antibiotics. You didn't
Yes,
use
we
anti-factor at
use
the time.
antibiotics, and the after this after a few years, we were developing the anti-factor lipids to use to repair mitochondrial function because we knew that we needed to have mitochondrial function for the full repair process to occur. We didn't really think of it in terms of removing chemicals at the time, but I just surmised that, these are very hydrophobic molecules. They range in membranes and lipid globules. They ought to be capable of partitioning these hydrophobic petrochemicals that are very toxic and very damaging. So we came to that later. But first, we were using it as to help repair so that the toxic materials, long after the infections are beat down, didn't persist.
Do you
So the
think
recovery process is very slow. We use these to help in the recovery process.
so? Do
Then
you
we
think
started using them for the chemicals. So we use them much higher dosage levels for chemicals for a much longer period of time as well. Because sometimes it takes a year to detoxify somebody using the membrane lipid replacement. But again, there's a benefit to that because we don't uh, we don't cause chemical poisoning in the process, so we can
Mm.
eliminate that as a side effect of the
So
detoxification.
do you think if you had the access to anti-factor, let's pretend, and obviously you can't because you had to develop it because you saw the Gulf War illness, but do you think just taking it alone could have solved the problem, or did you think you had to use antibiotics? Like if you saw someone now who was injured, say by COVID,
Well,
could they
it's
just simply use anti-factor, or do
it's
they need?
all it's all the numbers. It's all in the numbers. And the fact of the matter is, you probably can't take enough of membrane lipid replacement to prevent some of the things that happen when, for example, you get in a microorganism by inhaling it or getting it through a cut or infection or through your gastrointestinal system. Uh, so we don't know about the prevention of it, but we do know that your immune system has to be maintained, and this will boost your immune system and help maintain it. So in that regard, it will help repel infections and so on. But mostly we think due to not activating, but rebuilding, repairing your immune system and keeping it at peak performance.
Mm.
So
So
that's what's
you're looking
important
at
there.
more as a repair restoration function and the detox being sort of secondary to
Well, it's both
its both.
because you have to do both. You have to start doing both, essentially. What I'm saying is that detoxification is a much longer term
Oh,
type
okay.
of therapy. That's the difference. You treat with antibiotics, it requires some time. Matter of fact, that's one of the controversies that we got involved in with the Gulf War veterans because we found out because these microorganisms are very slow growing, that we couldn't just give a short cycle of antibiotics for a couple of weeks and so on and then stop it. That didn't help at all because they came roaring back
Mm,
because their division rates were so slow that we needed to use it for at least 6 months was the minimum of therapy. Most people required almost a year of therapy, and of course, infectious disease specialists were yelling and screaming at me. That was antithesis to their whole training and background, and so they thought I was a charlatan. And what am I doing trying to
yeah.
say that people should continue long term on antibiotics? But it worked. It just took a long time. But if
Yeah,
you think
we're
about it, it
dealing with
makes total sense.
we're dealing with something that's not natural. So I'm sort of interested in how we are combating that. Like obviously, if you're taking things like antibiotics, antivirals, and
Well, even
all
if
of
you're
that, it's a
talking about, for example, say a bioweapon, they don't just invent those bioweapons. They have a starting point for all of them. The starting point is generally a known microorganism that is a pathogen that causes a certain amount of damage, but it may not be very effective in terms of a weapon. And so they juice it up.
mm,
They juice it up by introducing certain genes, for example, or by selecting for certain subspecies or subproperties and so on, or both, to eventually get a highly pathogenic substrate that can be effective. And along the way, what they want to do is they want to make sure it's survivable, because if you use it as a biologic weapon, it has to be more survivable than the original native form. So you do things like make it heat resistant, for example. So you introduce different genes to make it more heat resistant. You make it sort of resistant to immunologic surveillance. So you introduce certain antigens, for example, that or certain other functions that immune suppress an individual. So there are all kinds of different ways of which these various
HIV
characters that
inserts
work
and
in this
things.
area have done to what they say weaponize a microorganism. Because not only do they juice it up to make it grow better, but they have to make it survivable. They have to make it uh, escape the immune system, the immune surveillance, so for example. And so there are a number of things that they have to go through to weaponize it. So now with the ability to modify genes and so on, it's much easier to do it than the old days, which required years and decades even of selecting over and over again substrains,
Mm.
subspecies, to eventually get to something that was very pathogenic. Now you can do it virtually in a matter of weeks. You could develop a new strain just using molecular biology to change the genes. So anyway, that's
So in terms of
the world we live
countermeasures in order to stay healthy and survive, NT factor is something that would help us with our mitochondrial function and to maintain our
It does.
barriers and
So,
integrity. And
so for example, just the common infections that are out there, influenza, COVID, and so on, they're just, floating around because they're respiratory infections. So what do you do to maintain effectiveness against that? Well, first thing is your barrier function. In the case of those are respiratory infections, so it's your epithelial barrier in your lungs. You have to maintain that proper barrier so you don't gain, those infections don't gain entry into your system, don't gain entry into the epithelium of your lungs. So the anti-factor lipids helps there because it helps maintain the barrier. So what does it also do? Well, it helps maintain innate immunity, which starts with your mitochondria. So that's important in terms of fighting those infections. It also helps in maintaining the other forms of your immunity as well, because it helps maintain the cellular systems that are responsible for those forms of immunity. So it helps with that as well. So it helps maintain your energy, which is important to resist those types of infections, even if they get inside your body. So all these things are all intertwined and all important. It also has another important property I didn't talk about. It helps transport important nutrients into your system. So it turns out that there are important nutrients, whether it's CoQ10 for your mitochondria or different types of things which are hydrophobic in nature, they're hard to transport. It encapsulates them and makes them easier to transport into your system. So it turns out that taking the anti-factor lipids helps in bringing in those useful cofactors and other nutrients that are absolutely essential for fighting off disease as well as maintaining your normal system. So it has just a number of different possible ways in which it helps you. That's why I think membrane lipid replacement is so important. It doesn't just have one way of helping you. It
yes,
has several ways of helping you. And they're endless actually when you think about all the different systems in your body that have to function properly. All your different organs, all your different tissues, all your different systems have to be working properly in order to fend off disease and to heal yourself from disease when it occurs. So that's
so
why this is so
ideally we
important.
don't want to get infected, and otherwise that's when the disease process starts, and it starts damaging these phospholipids in our body and leads to a breakdown
Yeah.
of the membrane. This whole adage of like getting sick, this idea that getting sick is a good thing, but it isn't, is it? Like what you're saying here, it's not. Let's let the body be at such a state where it's eliminating pathogens as fast as it can, so it doesn't have to repair it.
Oh, it has to repair.
I mean, it hadn't been
I'm
like it,
not
so it
saying,
doesn't have to repair
so
as much
people
damage.
ask me, well, why do you say that this is an adjunct therapy? Well, because in a way, it's not fast. So if people get sick, for example, because of a viral infection, you don't want to take them a year before you can see the effects. You want something that operates pretty quickly. So you want an anti-transcriptase. You want various immune functions. So you want them to be immunized. So you want all this stuff, but to get it to all work properly, you need membrane lipid replacement as well to ensure that it's working properly. And then again, you need the membrane lipid replacement to repair the damage that's done because of the
Yes.
infection and to allow the recovery, which is very important, to occur. And that takes time as well. So all these things turned out to be important. So that's why we say it's an adjuvant therapy. We don't want to interfere with the therapy that anybody's using. I'm not saying this is a panacea. It's not. What it is it helps in the maintenance, it helps in the repair, it helps in the recovery, it helps in the resistance. So all these different things are important when it comes to fighting disease. You can't say, well, we're going to only do one and not the other. No, I say bring it all on. This is just one piece of the puzzle, but we think it's an important piece.
And it's interesting you're talking about autism, and I'm going to sort of link it into Alzheimer's and the increased prevalence of it in our society. Do you think that that's a result of our increasingly toxic society?
Well, I do think that various toxins, chemicals, biologic, and so on, have a role in ASD and autism spectrum disorders. Now, let me tell you some of the background on that, because I've done a little work on this in the last couple of decades. And we started here in California with the Gulf War family, because we found the children were getting autistic. That was the tip-off that autism was a probably complex process that involved not only environment, but involved an infectious process, at least in many of the, maybe not all, but we think most of the autistic children. And we could see this in the children after the veteran who was exposed came back and passed the illness to the family members. We could see this happening in the family. So
I mean, that's pretty shocking,
in
isn't
that
it? The fact
case,
that they come back when their children become autistic.
yeah,
Because
let me continue, because they started with the Gulf War autism cases. So this is what led me into talking with families and also with some of the professionals in Southern California. This led me to start doing studies on something that wasn't related in any way to the Gulf War family. So we started to work in some rural counties in Central California, where there's a lot of farm work that goes on. These are counties where farming is like number one activity. Like in Salinas County, up in Central California, it's one of the top growing areas in the United States. So the entire county is practically farms producing lettuce and artichokes and you name it, any kind of vegetable you can imagine are grown there. So they use a lot of pesticides and herbicides in those. So it's interesting. Pesticides and herbicides are almost all backbones, hydrophobic molecules, multi-chain, complex molecules which get in and interfere, for example, with the nervous system of an insect or some kind of pest, and the herbicides which interfere with the growth of nasty sort of weeds and things like that you want to eliminate. So it turns out those hydrophobic molecules are concentrated in the normal vegetables and other things that you grow as well, and they're in the environment, so they're present in high amounts. So these children were running through the fields to go to school, for example, taking the shortcuts to school and so on, and they were showing very high rates of autism. So they were getting exposed to the chemicals up there. So we figured that maybe that was a problem. Turns out they were also being exposed to some of the same types of infections, not the same exact type, but similar classes of infections that we found in the Gulf War veterans. So although we found a lot of other infections as well in the children, but we did find a lot of mycoplasma in the children, just like we found in the Gulf War veterans and just like we found in the children of the Gulf War veterans. So mycoplasma was an important infection that we found in these rural counties and so on. But we found in the case of infections, they just didn't have one infection. They had multiple infections. They had a lot of different infections. So this is why it became a really complex problem. So this is, I published in a couple of neurologic journals, the fact that we found a lot of these infections in young children that were autistic and so on. And it turns out that the membrane lipid replacement, very safe for children because it's all in your body anyway. And so we could use this. It wasn't a drug. And so families could use this in their children without fear of poisoning their children with this type of therapy. So we use it a lot for the autism cases, and we found some good benefit from it as well. So that's the story of getting into autism spectrum disorders with it.
So were you able to, what sort of improvements were you able to see in the children who took it?
Yeah, no, the children, I don't examine the children. I can't do that. I'm not a statistical child psychologist to examine these because these children have a lot of different problems, but some of the learning problems and some of the social interaction problems and so on that we see in ASD patients have to be analyzed by a child psychologist. There's really no other way around it. So in the papers that I've done, just we've worked with those, we've worked with the families, we've worked with the child psychologists and so on, so we can examine some of the problems that were there. Yes, we did find that these families benefited, the children benefited from the membrane lipid replacement, but they also benefited from the other treatments that we were giving, for example, fighting the infections and so on. So again, it was a multi-step process. It wasn't that any one thing alone was solving the problem. So to get back to membrane lipid replacement, this is why I say it's adjuvant therapy for most
Yes.
any other type of therapy that you can imagine. So myself, I couldn't imagine anybody getting any kind of therapy without taking membrane lipid replacement because practically everything you do, whether it's injury, whether it's a gunshot wound, and by the way, we dealt with a number of those from the Gulf War, wounds that wouldn't heal.
Yeah.
And we were able to get those wounds to heal if we added in membrane lipid replacement into all the therapeutic regimens that they were already employing. So again, it was an adjuvant to things that were already being done. So we knew that this was really important. So infection, tissue damage from disease, for example, and anything from cardiovascular disease where we know that lipids are involved to, for example, we're now working with severe mental illness because we know that lipids are a factor in severe mental illness. Because why? Because when you do lipid profiles of people that are bipolar or have schizophrenia or something like this, we find abnormalities in their lipid profile. And how can we correct those abnormal lipids that we find? Membrane lipid replacement.
Mm.
So I'm trying to organize a trial at a lot of the California state hospitals that deal with, for example, these severe mental cases of people, for example, if they're institutionalized with a severe mental illness. This is a group that I can work with because they're not going to go anywhere anytime soon. And so as long as I can work with the hospitals, we can test these out. We're just trying to get a protocol approved for first one. We're going to look at is schizophrenia. So yeah, it's got a lot of different uses.
And what have you seen with Alzheimer's? Have you seen improvements there?
Okay, so for neurodegenerative diseases, which is really quite different from the severe mental illnesses in terms of the etiology and what's involved here. There's a lot of damage that occurs in the neurodegenerative diseases, and so the empty factor lipids help with the repair of that damage. It also may help with other factors that are involved. For example, in my studies on Gulf War illness, I got involved in looking at infections there because we found there's a very high incidence of certain neurodegenerative diseases in the Gulf War veterans. For example, ALS, Lou Gehrig's disease, very high incidence in Gulf War veterans. And these were all very rapidly progressing diseases. So we had to work fast because a lot of these people died fairly quickly. So what we did find is if we used antibiotics and the anti-factor lipids, because all these 100% of them were mycoplasma positive,
Yeah.
we could slow that process down and so on. But it turns out we got to these people too late because it was more than a year or so after they had the onset of the disease. So we're already dealing with late phase diseases. So we were able to stop the progression, but unfortunately, many of these people went on eventually to pass away. But we were able to stop progression in some of the most severe cases, and that was heartening. We felt that if we could get to these early cases, we might be able to do something.
So what about now, like in terms of people being diagnosed now with Alzheimer's, do you think that there's hope to get
I think, again, as I said before, I think there's a place for this in terms of adjuvant therapy because there's place for healing. For example, the fibril tangles inside cells, the tau malformed proteins, and all these things have to do with cell damage. They cause cell damage. It needs to be repaired. And so the anti-factor lipids help repair that cellular damage, but they also may slow down this process of forming these abnormal structures within cells. And we're interested in that. We haven't found the right, combination of researchers and clinicians and everything to make that happen, because people working on different aspects of it, they only want to work on that aspect. So it's hard to get them involved.
But it's very interesting, really, when you think about how many applications this has. Literally, everyone should leave hospital with some anti-factor, right? Even before they get into hospital, they can use it to, enhance their health. But it's not, as we discussed before we started this, it's not very well known. Like I don't think I've heard anyone talk about anti-factor within my, circles. And why do you think that is?
That's our fault. We need to communicate better. I can only do so much myself, but we
Yes.
need to spread the word. And this is one of the things I keep telling the company that makes this product. I keep telling them, we've got to get out there. We've got to have more communication. So this is why I hope doing your podcast and any others I'm willing to go on to help spread the information, because the more information gets out there, the more people see that, hey, this is useful. Even as an adjuvant, I can continue to do my own studies on my own thing, but adding this onto it could enhance the outcome that I'm trying to achieve. And so this is something we have to do. So we've used this for cardiovascular diseases. We've used this for neurodegenerative diseases. We use this for chronic infections. We've used this for trauma. For example, I mentioned gunshot wounds, but also there's a lot of trauma in, for example, in a heart attack, there's a lot of trauma. So we've used it for that. And it turns out it's being used for things like heart attack in Eastern Europe, where they give IV membrane lipid replacement immediately after a heart attack. And turns out in their studies, it's very important for recovery from a heart attack, from heart ischemia. And so we're doing that. I actually have experiments going on in Montevideo, Uruguay, where we're taking beating animal hearts in vitro, and we're causing blockage of heart vessels, and we're seeing in real time what happens with a heart attack and what happens if you put membrane lipid replacement in before, during, after, or various times. We can do all the different combinations and show what is the best time to apply membrane lipid replacement to help them recover, help those hearts recover from a heart attack. So that's ongoing at the moment.
It's very exciting, really, when you think about the possibilities that it has. I mean, even when I've looked at the study that you sent about the heart. I mean, these things are measurable. You can measure people's blood pressure, their lipids, HDL. All these blood markers are improved by anti-factor, right? So
absolutely. I'll give you a very personal example of many. I'm going in to have my lens replaced in my right eye in about a month, and if the conjunctive damage, or if the damage hadn't occurred over the years, the macular degeneration, so on, in that eye, I wouldn't have to have the eye surgery that I'm having to have. So cataracts were my primary problem there. So some of this may have been preventable if I'd taken anti-factor lipids 40 years ago, let's say.
Yes.
So it's just something that requires a long-term solution for a lot of the things that ails us.
Even though in our modern sort of toxic society, with all these things happening around us, there are people working on solutions. And I think this is a very important thing to put out there, because, a lot of people are going to kind of a state of despair, I guess, because they're seeing so much illness and death around them, that, that you've been here for decades working on solutions.
Well, these are solutions, but they're not short-term solutions. They're long-term solutions. Yeah. And that I have to warn people that this is not a quick fix by any means. Everybody wants a quick fix. No, this is a long-term recovery process. Is better explained.
But it is one. It is something that can assist people to heal.
Oh, absolutely. We've proven that. We've shown that. It helps people. There's absolutely no doubt about it. For example, I'll give you the example I was talking about, heart function. we've taken people that have that are at very high risk for dying of a heart attack. They have homocysteine levels in the danger zone. So homocysteine is a molecule that is a marker. We don't know its exact involvement in the process of heart disease, but we know it's a marker because people at their homocysteine level, we can predict if they're going to have a heart attack just on the levels of homocysteine in their blood. So we know that this is a great marker. We know it can predict heart attack. We know it can predict hospitalization. We know it can predict death. So what happens if we take people that aren't dead yet, haven't had a heart attack yet, but have homocysteine levels in the danger zone? It brings it down. Long-term membrane lipid replacement with anti-factor lipids brings that high homocysteine level down in the safe zone. And guess what? Those people never went on to have heart attacks. So we know that as a preventive thing, this is incredibly useful.
Do you think, as a theory, misfolded protein could be seen as mitochondrial misfunction? And
Oh, absolutely. Well, if you take, for example, misfolded proteins, because this is incredibly important in neurodegenerative diseases, just as for one, but it's also important in aging. It's important in a variety of other different diseases as well. So when proteins get made, they get made on a lipid bilayer matrix in the endoplasmic reticulum, and that the property, that physiologic status of the matrix, is very important. It turns out for the misfolding that can occur during the synthesis of protein. So I think it's very important in those misfolding protein diseases. So, but to prove that, we've got to get people on membrane lipid replacement before they have the disease. So that's not so easy when you think about it.
yes,
But I do think for people, if they're concerned about disease, if they're concerned about aging, if they're concerned about resistance, if all these things, they ought to be on membrane lipid replacement.
it's part of a holistic framework. I would see it as, and very good in terms of if you've got something like long COVID. I mean, there's nothing that you can do that you can go wrong. I mean, obviously, people have to take it for a certain period of time.
You know, it's very unfortunate that people that do holistic medicine have got the bad name. They've gotten ridiculed from their colleagues, which might work on different types of medicine, but more short-term solutions for medicine. And so homeopathic medicine is, I think, absolutely essential. And that's really what we're doing with anti-factor lipids is holistic medicine, because it's really looking at some of the basic research, basic biology that occurs in the disease process. But the basic pathogenesis of a disease, you're talking about to prevent it is you need a holistic approach. So there's nothing unusual about it. It's just that, I don't know, people don't consider me a maniac when it comes to eating green lettuce or things like that. I mean, it's because essentially all we're doing is using basic science to try and explain some of the homeopathic mechanisms that are so important, and then seeing what can we use with natural molecules to prevent this, to reverse it, to whatever. So it's really, basic research driven homeopathic medicine.
So with anti-factor, anywhere that it says anti-factor is a patented process, you can be guaranteed that that is the same product that's been used in the clinical trials. Is that correct?
Yes.
So it doesn't matter who sells that particular brand of product.
Well, there are a lot of different products out there that people put in the similar types of lipids in them. But unfortunately, they either don't put enough in, or some of the ones they put in are damaged already.
Okay.
And they put them in. They're not protected, is the other major bugaboo. And so the shelf life is very short. And they don't really worry about that, but it's very important. And so there are a lot of things that are going on that you have to pay attention to when we talk about membrane lipid replacement. So this is why we've thought about a lot of these problems. And so with anti-factor lipids, we've solved a lot of these problems. But even then, we occasionally run into a problem of a bad lot, for example, where there was some process in the manufacturing of it where they made a mistake or they didn't do something right. And so we ended up with a bad lot. So we have to throw that out. And so you know, you still have
So,
to
I
be
mean, how
careful.
did you even think about producing it? I mean, like, it's a soy-based product, yes?
Well, we have it from different sources, all plant.
Yeah.
So soybean, for example, is another source. Soy is a source. We had a problem with soy, even though we don't have any proteins or glycoproteins in the product. It's all lipids, and there are no lipids involved in soy reactivities. Or people have soy reactions, allergic reactions. We don't have that with NT factor. We've taken people that have extreme soy sensitivity. They don't have a problem with the lipids because basically the lipids are the same. But if you don't make it properly and you get some soy proteins in the product, final product, you're going to have the same sensitivity. So you have to be very careful how the product is made. So we have a sunflower product, so there's no soy involved in that. And people that have soy sensitivities, they like that product better, even though it's more expensive because the source is more expensive and it's more involved to make it. The yield isn't as high. So, but most of it's made from soybeans, and most everybody doesn't have a problem with that.
So what sort of dosage should people be taking?
Well, it depends on their problems that they want to solve. If they're interested in anti-aging, 2 to 4 grams per day. I
Yeah.
take 5 grams per day myself. That's a little more than most people probably need to take for anti-aging. I'm 82. People tell me, you don't look 82.
You're very
I'm
sharp
glad.
still, yeah.
I'm glad. So, but I'm going to take it the rest of my life. So on. Unfortunately, my father was like 93 when he started. And he was in one of the first clinical trials because I got him enrolled in one of the first clinical trials for anti-factor lipids. And just like everyone else, we measured his mitochondrial function, we measured his fatigue level, and so on. He benefited quite dramatically with it. And he went on to live, he lived to 97.
Okay.
And, but I think if he'd gotten it earlier, maybe he would have lived to 107. Who knows? So, but he benefited. The important thing was, even at 93, he benefited. And the interesting thing is that when we looked at mitochondrial function, we were able to take people in their 90s and return their mitochondrial function to the level of a 30 or 40-year-old. So that in itself tells you that this stuff is really great as an anti-aging tool.
So it's quality of life as well. So how did you go with the Gulf War veterans that you were treating with anti-factor? Are they back to living a normal life?
Well, most of them, again, they suffered for years without any treatment whatsoever or any useful treatment whatsoever. So we came late on the scene there. And unfortunately, if I'd gotten to them when they were much, much younger, we'd have gotten much better results. But they tell me the feedback is that they're doing great. And these are people that, remember, had Gulf War illness for a decade or more before they ever went on membrane lipid replacement, some 2 decades before they went on membrane lipid, and they still got benefit from it. In fact, my chemical exposure group from the Gulf War, these are all people that were essentially untreated after the Gulf War, and they'd gone on for let's see, 25 years before they were treated, and yet they got very dramatic and importantly, statistically significant improvements in their condition.
I mean, after all, the body does really want to heal itself, given the right ingredients and
It
time,
does want to
right?
heal itself. It does try, but you have to give it the tools. And this is what we think we're doing. We're giving it the tools. The mechanisms are already there. They're already built into us. Our survival mechanisms are there. We just have to make sure that we give it the right nutrition to keep it buzzing along at maximum functionality. And that's basically what we're doing. We're
Mm.
just improving the function that's already there.
And it's amazing because there's nothing to worry about in terms of side effects.
No, this is one of the interesting things. So anti-factor lipids have been designated by the US FDA as a GRAS supplement. GRAS stands for generally indicative of safe use. Essentially, that's GRAS, but so people can use it with confidence.
Yeah, and so IMED is primarily a research institute. And so if
Yes,
people, if
it is a
doctors
research
wanted
institute.
to, or researchers wanted to approach you about any clinical trials or anything like that, that's where they would go to the imed.org website. Is
Yeah,
that?
or they could contact me by email. My email site's on the website. Um, I was getting so many. I'm in my home office right now because I've been retired for years. I'm newsflash. I'm 82 years old. I'm still not. I've been retired for years, but I'm still running pilot clinical trials. I'll run the pilot and turn it over to somebody else to really do the hard work, which is really to organize a RCT, randomized controlled trial. I just can't do that at my age.
But
I'd
it's
like to do
amazing,
things
really,
myself.
isn't it, that something like this, it's almost, to me, it's like, do we test that water hydrates the body? I mean, like, it seems so logical to have a product that is, you can take that will repair the cell membranes. How many studies do we need to prove that it works?
Well, we've done 7 different clinical trials, for example. Most of these were pilot. We've done a few more pilot studies that I've published in case report journals, for example, just to try and get the information out there so that people can see that it does work. For example, the case of gunshot wounds. I don't have a full publication of gunshot wounds, just as a case report that describes how to use it for gunshot wounds, but that's probably sufficient to get people to use it, to try it.
Mm,
And so, some of these things, because we're dealing with a nutraceutical, we don't always need a
yes.
full-blown, year-long placebo-controlled trial to show its usefulness or utility. And the fact that it's so safe, one of the main reasons you do a clinical trial is for safety issues. Well, this doesn't have safety issues, so we don't have that problem.
So it's mostly getting the word out there, because ultimately people can just pick it off the shelf themselves
Yeah, it's getting
and
the
use
word
it. So
out
if
because
they've come
people,
back from hospital, or if they've got COVID or
yeah,
whatever their illness is,
absolutely.
Alzheimer's, there's really no risk to take it and all benefit.
Yeah, most people have stumbled across it by word of mouth, for example. Because the company is so small, it's still a tiny company that makes this. And they haven't had the resources to go out in a big campaign, to make it the new aspirin. This would really require a multimedia campaign. That's very expensive, turns out, to
Yeah.
inform the general public. And so what I've just been doing is slowly but surely just informing a few professionals here and there, and hopefully they'll inform their patients and they'll inform other people, and the word will get around. But word of mouth is not a very rapid or effective way to do
No,
a marketing program.
not when we have internet censorship like we do. And I think too, it it the more sensationalist products are easier to market, and the more,
Well,
you're
that's
marketing it as
exactly
a
what I
very
found.
adjuvant. It's a
That's exactly what I found.
there's nothing very sexy about advertising for it. So it sort of drops to the bottom of,
Yeah.
the rankings and
And
everything.
if we did come up with a big advertising campaign, the people that we really want to reach would say, "Oh, that for sure has got to be bad because otherwise, why are they spending all this money for advertising?"
Yes,
So we're
side.
trying to figure our way on how to what's the most appropriate way to do this. But along the way, I'm just trying to get professionals to use it and their patients to use it, and hopefully it will spread and the word will get out in a good way, not in a bad way.
Word of mouth. Definitely. And I think that's how things are becoming these days. It's about, who do you trust? Like, I know you've quoted these organizations, but public trust is so low. But when, this is why I think it's very important that you've done this interview today, is to, you're putting your name and your reputation and your background, the fact that you've survived, you know, through this whole biowarfare era, to your age, on the line to endorse a product that you've developed. And I think this is an excellent way to let people know about it and to give it a go, because ultimately there's only all benefit and no risk. It's either going to work for them, and it will, I think if they stack it on with other things that they're doing, that this is where we've done a lot of interviews on holistic healing. If you just take the totality of that thing, with nutrition and sunshine, exercise, and all of that, I think people will be able to achieve optimal health and live for a very long time.
Yeah,
and,
and I
and
recommend
healthy.
all those things, by the way.
Pardon?
I try and on my phone, I've got, my miles walk every day. Uh, Try to eat good food, eat healthy food, all that sort of stuff, and take the anti-factor lipids on top of that. So I'm
Yeah.
a walking, talking example of that.
Yeah, you definitely are. I mean, at 82 years old and still sharp as a tack, um, you're a living
Well, I don't know
example.
about that. Some of my students would probably say, oh, wait a minute there, maybe not quite as sharp as he used to be.
Yeah, well, everyone gets old at some point. Who knows, maybe you need just some more NT factor.
Well, I've stepped it up. As I get older, I've taken more, actually.
Yeah,
The
because
reason for that, really, because it turns out that it's harmless, essentially. You can, there's studies that have been done when the FDA approved it. There were studies on phospholipids of similar types that not the entire thing that we do with the protection and everything, but just phospholipids themselves. They were taking 40, 60 grams per day. Well, the maximum we've used in a clinical trial is 6. So we're way below
is there any
the
foods
control.
in particular that you focus on
Beg
for long?
pardon?
Is there any foods in particular that you focus on?
Oh, no, I try and eat vegetables every day, and I try and, eat healthy fats like olive oil. And, but basically I don't, I'm not that picky. But there's certain things I try and avoid. Processed foods, I guess, I try and avoid. But other than that, most foods are okay by me.
Yeah, because you can repair the damage, I guess.
Yeah,
Yes.
well, I know I'm taking the anti-factor lipids. I know I'm going to be okay.
Okay. So anyway, where is there anything else you would like to add? Otherwise, I
Well,
can.
I'd like to give people the website,
Yes, we'll put all the links.
ntfactor.com. Very
Yes, we'll put all
simple.
the links to the bottom of the and on our page. So ntfactor.com and
Yeah,
imed.org.
right. immed.org. So those are two places they can go for information. I put a lot of sort of simple, easy to read things on the antifactor.com website. I've done this last year or so. I've updated them, and so I've got 13 brief articles there that covers everything from um, weight loss to aging to uh, chemical exposures to, you name it. We run the gambit on there, to mitochondrial function, to fatigue, to sleep dysfunction, all these things that we've found that NT factor helps.
Amazing. And we'll also put some links to your books and your other YouTube interviews. So literally there's
Sure,
hours and
great.
hours of information that people can access. I think this has been a great introduction for them about the product and about your.
Yeah, and if they want the basic information, they could go on the ntfactor.com website, for example, and I put all my publications in this area on that website. Obviously, I'm not going to put 700 publications. I put the 50 most important publications about anti-factor lipid on there, and they could download them for free because I made sure that most of them were published in open access journals, so people can get them.
Yeah. So we'll post all the important links related to this podcast in the description, as well as this hot class will be available on YouTube, Rumble, BitChute, on our website, on LinkedIn as well. In closing, I wanted to remind viewers to check out our other podcasts, which do all tie into each other. Please post all of these in your groups, in your chats, send them to friends and families. The algorithm actually does not favor quality content because it generally favors clickbait. So you can also find a list. So if you wish to support our work, you can do so by visiting the donate page on healthallianceaustralia.org or also join our free mailing list. We are very active on LinkedIn, so please join us there. Thanks everyone for watching, and thank you, Professor Nicolson, for your time today. It was a wonderful interview.
Thanks for having me.